It is my pleasure today to introduce you to Dr. Ellen Francis, who is a postdoctoral fellow in the Life Course Epidemiology of Adiposity and Diabetes Center, currently funded by her K99R00 grant to characterize metabolic heterogeneity in pregnancy using metabolomics data. Prior to joining the University of Colorado, Dr. Francis completed her dissertation as a predoctoral fellow at the National Institute of Child Health and Development in the Department of Intramural and Population Health Research Epidemiology branch. Broadly speaking, Dr. Francis is interested in risk factors of perinatal health and the consequences of these factors on obesity-related disease in offspring, with a recent focus on the relationship between novel metabolic phenotypes in pregnancy in relation to placental function and early childhood metabolic health. And so I'm going to turn the floor over to Dr. Francis and ask that you all hold your questions or enter them into the chat, and we have some time set aside at the end for Q&A. Ellen, whenever you're ready, you're already sharing your slides. I'm going to mute myself.
Unmute myself. Great. Thank you so much, Dr. Pern. I'm really excited to be able to share not only a little bit about my work, but a little bit about who I am. Even though I've been a postdoc at University of Colorado for almost 3 years now, I think I haven't had many opportunities to sort of present where I came from and what I've been doing and kind of where I see that going in the future. So I'm going to pull up my laser pointer. Here we go. So I have a background in psychology, and then I went to work as a research associate and data analyst in a behavioral health organization for about 5 or 6 years. And the reason I think I find it important to bring this up is because it determined the type of PhD I decided to go and pursue, which was an applied health research and evaluation doctoral degree. And essentially what that means is that during my coursework and my training, I was introduced to a variety of analytical techniques by which you can answer health-related questions. So qualitative research, mixed methods design. I was trained in health services research from econometricians, and of course, epidemiology. And when I was introduced to epidemiology, it was really the first time that research was presented to me in a way that you could systematically organize not only your question, but how you were going to answer that question, and then the relationship of variables that were important to investigate. But given that my program was sort of painted in broad strokes, the variety of methods that we were taught, I went and applied for a fellowship at the NIH to finish my doctoral research in the epidemiology branch at NICHD. And coming to the Lead Center was really a very nice natural extension of my PhD work, because I was interested while at NIH in pregnancy-related metabolic conditions such as gestational diabetes and the consequences on fetal growth measured and assessed at delivery. However, the outcomes that I'm interested in offspring tend to emerge over many years. And so coming to the Lead Center, where we have longitudinal follow-up of kiddos, was a very nice next step. So I see my research interests sort of falling into 2 spheres. One is in perinatal women's health, and I'm interested in this sphere within obesity and diabetes and diet. And the second is in child metabolic health, and particularly insulin resistance, and a little bit of child development as throwback to my days in psychology. And I see these 2 spheres overlapping in this concept of developmental overnutrition, which I'll talk about a little bit more in detail later on. And since coming to the Lead Center, I've been trained in framing my research questions using a life course approach, which essentially means that I look at the different physical and social factors that operate at different life stages that affect chronic disease, with the aim of identifying the underlying processes that operate across the person's lifespan. And I really like to show this graph or this sort of depiction of how that gestational environment in particular can shape health across the lifespan that Dr. Pern put together, because it highlights my interests very nicely. So I'm interested in obesity and diabetes in pregnancy, which can affect a myriad of different factors of the maternal fetal milieu. But particularly, I've been focusing on this transfer of maternal fetal fuels. And we tend to think of those as compounds such as glucose or amino acids or maybe fatty acids. Excuse me. I forgot to mention that while I do not have COVID, I do have something else going on. Okay, so interested in the transfer of fetal fuels. And that interest really started during my days at NIH, where I was part of a team building a biomarker risk prediction model for gestational diabetes assessed in early pregnancy. And my sphere in this team was really to focus on adipokines, which are peptide hormones secreted mostly from your adipose tissue. And they've been involved in different facets of glucose metabolism, such as improving insulin sensitivity. And what I found during this work was that some levels of these adipokines, such as the soluble leptin receptor or adiponectin, were related to a reduced risk of developing GDM later on. But beyond just looking at this association, I wanted to see if these adipokines would improve our ability to detect which women developed GDM. And so I added them to a conventional risk factor model. And these risk factors are things like a family history of diabetes, pre-pregnancy BMI, and even circulating levels of glucose. Oh, here we go. And what I found is that if you added levels of these adipokines to this conventional risk factor model, we saw a slight but significant improvement in the AUC. Now, I want to point out that an area under the curve value of 0.7 isn't terrific, and an improvement from 0.7 to 0.74 isn't mind-blowing either. But what this did do for me was plant a seed that I think has been sort of unconsciously growing since this time, in that there are factors of a woman's metabolism other than just those traditionally related to glucose that are implicated in her route to develop high blood sugar in pregnancy, and also just sort of affirmed this interest in the relationship between glucose and adiposity. And these differences in GDM related to insulin or other factors involved in glucose metabolism have been studied by other researchers, and I'm showing some work here by Camille Powell on different subgroups of GDM based on insulin physiology. So for instance, on the left, we can see there are different groups of women based on their insulin signaling who have relatively similar levels of glucose in response to an OGT. However, given their difference in insulin signaling, they are diagnosed with GDM because, for instance, this woman in blue who has an insulin secretion level similar to that of a woman with normal glycemia is insufficient to meet the demands of glucose. And what's interesting to me is that these different subgroups of GDM, these different routes by which you can get to hyperglycemia, are differentially related to risk of perinatal outcomes. So for instance, the red bar represents a woman who potentially has an insulin resistant phenotype. And we can see that they are at increased risk for their offspring being LGA, pregnancy-related hypertension, or NICU admission. And although this is a little bit outside the scope of what I'm working on and presenting today, it's not just that one subclass of GDM has an increased risk of all poor outcomes, but that the risk is dependent on the outcome of interest. So for instance, the women with a deficiency in insulin secretion or a mixed physiology have increased risk for polygenic, or an increased polygenic risk score for type 2 diabetes postnatally. Excuse me just for 1 second. Okay. And one thing that is important to realize is that the risk associated with exposure to gestational diabetes or hyperglycemia in pregnancy isn't just related to the woman, but also to her offspring. And I like to show this slide and this work by Dr. DeBellia that just nicely demonstrated the specific impacts of the in utero exposure to hyperglycemia, such that offspring who were exposed to diabetes in pregnancy are more likely to develop diabetes in youth compared to offspring who are unexposed, and that's shown here in the red bars. But it's not just completely due to genetics or a shared environment. And this was also very nicely shown in a design in which youth who had siblings who were discordant for exposure to diabetes, meaning that one sibling was born before and one sibling was born after mom's diagnosis with diabetes, that the sibling born after had an increased risk of developing diabetes compared to their unexposed sibling. And so what this indicates is that the in utero environment is a really important time point in a offspring's life in which exposure to excess fuels, particularly, can result in differences in their predisposition to metabolic disease later in life. And that's what the hypothesis of developmental overnutrition indicates, is that these differences in adiposity and glucose homeostasis increase their risk of metabolic disease later in life. And many studies tend to focus on outcomes assessed at delivery. And this makes sense, and it's what I focused on at NIH myself. And it's because at delivery, it's most proximal to that exposure of overnutrition. However, chronic disease and metabolic conditions tend to emerge over a person's life course. And so looking at the metabolic health of a child in early life may be this window in which we can see how these fetal exposures in utero track across the offspring's life. And so when I was thinking about developmental overnutrition and this interrelatedness of adiposity and hyperglycemia, I was brought back to this relationship between insulin resistance and insulin secretion, and how there are different routes that you can get to hyperglycemia. So what this blue line is showing is someone who has normal glycemia because their insulin sensitivity is relative to their insulin secretion such that they are able to maintain normal levels of glucose. However, folks that fall along this brown line tend to be categorized as being hyperglycemic because, let's say, for instance, their level of insulin secretion, which might be up here, is insufficient to their level of low insulin sensitivity such that they develop diabetes. And we know that adiposity tends to overlap with insulin physiology, such that Someone with obesity and hyperglycemia tend to be grouped over here on the left and be classified as someone who is insulin resistant or has low levels of insulin sensitivity. Whereas someone who has a more lean BMI and hyperglycemia might have an insulin secretion deficiency. But in terms of pregnancy, one of the things I'm also interested in is how obesity in the absence of hyperglycemia also might impact the transfer of these fuels or developmental overnutrition. So I had two questions regarding exposure to developmental overnutrition, and particularly subclasses of developmental overnutrition, which I've defined as having obesity, GDM, or obesity and GDM. The first being, are the effects of a poor metabolic health, such as being obese or having GDM in pregnancy, detected in the offspring in late childhood through adolescence? The second is there a measurable overlap or difference in the effect of these different subclasses? Essentially, is there a difference between exposure to GDM alone, obesity alone, or GDM and obesity? To look at this question, I used data from the EPOC study, which is one of the sort of birth cohorts or longitudinal measures of prenatal exposures and offspring health here at the Lead Center, in which we enrolled kiddos at 10 years of age. And we retrospectively abstracted prenatal medical record data from moms for GDM diagnosis and pre-pregnancy BMI. And then we followed offspring up to 16 years of age roughly and took another blood draw. And then that blood was assayed for untargeted metabolomics. Now, to do this analysis, I set it out I'm going to present it in two major steps. So the first step was to conduct what's called an MWAS, or a metabolome-wide association study. And essentially, we screened all 637 annotated metabolites for an association with exposure to any overnutrition, GDM or obesity. Then we applied an FDR correction for multiple comparisons. And of those 52 metabolites that were identified to be significantly associated with any overnutrition, we brought those forward and then created profiles from those 52 metabolites using principal component analysis, which is a way to just reduce the dimension of the data to something that we can analyze. And then in this step, with those profile scores, we looked at an association with any overnutrition subclass. Again, that's obesity, GDM, or obesity and GDM. What this is showing is just a graphical representation of that second step. So after we identified the 52 metabolites associated with developmental overnutrition, we found 4 profile scores, and in these circles are the percentage of variation in those 52 metabolites explained by that profile score. And then the key compounds that I've listed out on the right of each of the 4 profiles represent the top positive loading compounds of that profile, which you can think of as being most representative of that profile. So for instance, this big blue square, which explained 44% of the variance, was this gamma glutamyl profile, and it had derivatives of glutamate, lysine, and glycine. For instance, this green square, which explained 6% of the variance, this profile was represented mostly by keto acid metabolites of leucine and isoleucine in the TCA cycle intermediaries. I then looked at the correlation of these profile scores with other markers of child metabolic health, such as body composition, lipids, physical activity. And I found that a higher score on the sphingomyelin mannose profile or factor was positively correlated with greater visceral fat. We also found correlations with lower HDL and higher energy expenditure. And so to get at the second part of my question, which are there differences or similarities in exposure to these different phenotypes or subclasses of overnutrition, I looked at the association between the profile score, and each profile is represented in a different box. So your gamma glutamyl profile is in this first box, for instance, and testing the exposure based on obesity and GDM, GDM, or obesity. And I found that there was no difference in this metabolite profile score based on whether the offspring was exposed to obesity alone or GDM alone or obesity and GDM alone. And while I did see some differences in the other metabolite profile scores, I just want to remind everyone that these profiles explain a relatively small amount of the variance in the data. So while we saw that developmental overnutrition was associated with metabolic changes in the offspring, I didn't find that there were substantial amounts of differences, suggesting that there's possibly greater pathophysiological overlap than difference. And this is not what I thought I was going to see. And so the premise for my NICHD proposal was really to kind of thinking about why it is that I didn't see the differences that I thought I would. And essentially, I sort of came to this idea that if I'm categorizing or classifying developmental overnutrition as this exposure to excess fuels, then what I really should be looking at are the fuels and not these proxies for the fuels. And the problem with looking at the proxies, such as BMI greater than 30 or GDM, is that it could result in exposure misclassification or maybe capturing some women into a overnutrition phenotype that shouldn't be. Now, if we could improve how we're classifying our exposure, it might assist us in assessing how much risk of offspring obesity or the related diseases are actually attributable to the gestational environment. And to sort of show this graphically, what I was looking at before was obesity, GDM, and obesity. However, there's a lot of metabolic information that is captured both within these dichotomous categories as well as beyond. And if we can capitalize on this metabolic heterogeneity, can we essentially improve our risk stratification in pregnancy? And can we use cluster analysis to do this? The Healthy Start study is a longitudinal birth cohort through the Lead Center in which the data lend itself to answering this question very nicely. So in pregnancy, we have 2 blood draws, and then we have a variety of lifestyle data collected and on physical activity and diet. And then at birth, we have body composition measured by DEXA, so we can get the percentage of fat mass along with birth weight. And then kids were brought back at about 5 years of age, in which we have another fasting blood draw and some lifestyle questionnaire data. And they're actually coming back for a third visit, but for the purpose of this talk and of my grant, the data ended at the 5-year visit. And so what I'm going to present now are the preliminary data that were in the K99R00 proposal to show that this idea of categorizing metabolic heterogeneity in pregnancy, I guess, has legs, so to speak. And so what I did is, with one of the blood draws in pregnancy, so the earlier blood draw, which was roughly in the second trimester, I used K-means clustering based on inputs of glucose, insulin, and the ratio of HDL to triglycerides to identify different clusters. And I picked up 4 clusters, which I've shown in different colors here on the left. And then to describe who these women are, I compared differences in their characteristics. And so, for instance, women in cluster 3 and cluster 4 tended to have older age. They also tended to have a greater educational attainment. Women in cluster C2 were racially diverse, and women in cluster C1 and C2 tended to be have more children, or to be less likely to be nulliparous, and they also tended to smoke more in pregnancy. Women in C3 and C4 also tended to have a pre-pregnancy BMI in the normal range. And women in C2 tended to have a poor diet quality, a greater prevalence of gestational diabetes, and to gain an excessive amount of weight in pregnancy per the Institute of Medicine guidelines. And so what you can see is that by using this clustering technique in pregnancy, we've identified some social determinants of health, as well as some differences in lifestyle behaviors during pregnancy. I then went on to look at how these clusters were related to differences in metabolic markers later in pregnancy. And I found women in C1 tended to have a higher level of triglycerides, and women in C3 tended to have a higher level of cholesterol. I also looked to see whether there were differences in offspring birth weight and fat mass at delivery. And I ran some pairwise comparisons and then corrected for that to help identify a reference group for further analyses. So what I found was that women in C2 and women in C3 tended to have a greater birth weight Z-score and a greater percentage of fat mass compared to women in C4, or offspring of women in C4. And indeed, women in C1 and C2 and C3 had a greater risk of offspring being born LGA compared to women in C4. Now, what these data are showing is that there are sort of meaningful differences in these clusters in terms of the maternal characteristics and in terms of some outcomes measured at delivery. But one of the things I wanted to address with the R00 portion was whether we would also see differences in placental signaling and also offspring metabolic health as other ways to quote unquote validate the importance of these different clusters. And so, partnering with Dr. Thomas Jansen, I was able to use some data from the placenta, and we found that cluster membership was associated with differences in placental signaling. So cluster 1 had a higher activation of ERK1. And so ERK is one of the functional readouts of mTOR, which is a really important nutrient transport system for the fetus. I also found that cluster membership was associated with a lower number of IGF-1 receptors. And this is interesting and aligns with another study that I did with Dr. Jansen, in which we looked at maternal diet quality and found that there was actually a greater number of IGF-1 receptors among women who had a higher quality diet. And if I can just remind the audience that cluster 2 was a group of women that were characterized by low diet quality. So this tends to make sense and align with what we've seen previously. Lastly, I looked in childhood and found that cluster membership was associated with greater risk of childhood obesity. And childhood obesity is categorized as greater than the 95th percentile of BMI based on CDC reference data. And then I had been interested in understanding if cluster membership would sort of give us more information than these other risk factors that we traditionally think of as being related to offspring obesity. And so I compared the magnitude of these estimates to those from exposure to BMI greater than 30 pre-pregnancy and GDM. And you can see that the magnitude of risk associated with cluster membership is greater. And it's also greater than other exposures occurring in the gestational, postnatal, and childhood periods, such as smoking in pregnancy, being breastfed less than 6 months, or the child's own concurrent total calorie intake or physical activity. And so what I'll be working on over the next probably 4 years or so is to kind of redo this analysis using different types of information. So we'll use metabolomics data that hopefully will be ready any day now to regroup and create clusters, or what are sometimes referred to as metabotypes, like a metabolic phenotype in pregnancy. Then assess differences in lifestyle based on metabotypes, and also looking at differences in traditional metabolic markers such as glucose or lipids. And this just helps us categorize what those metabotypes might mean, because untargeted metabolomics is really a hypothesis generating approach. Then we'll look at how those metabolic clusters are related to differences in placenta function, such as nutrient sensing, inflammation, cell proliferation. And then a little bit down the road, we will get metabolomics in offspring at that 5 years of age time point where we have fasting blood. And I'll generate metabolite profiles similar to what I showed in the EPOC cohort. And this is useful in and of itself to understand what metabolite profiles look like at this time point, but also as a way to assess how metabolites are related to lifestyle, such as diet, physical activity in childhood, other traditional metabolic markers, and body composition. And then to put it all together, to look at how metabotypes in pregnancy are related to offspring's metabolic profile. And then also to see how much is mediated by differences in placental function. And this all has implications for concepts of deep phenotyping, or precision prevention and intervention, which is a growing focus in my work. It'll also help us potentially create more robust comparison groups in pregnancy. And in offspring, it might help us understand and identify some underlying metabolic risk factors for early identification of later disease groups. So I see this work potentially being able to be replicated within some of the consortium. However, I want to see what what is most interesting about our findings. But metabolomics, I believe, will be available in most women in pregnancy within the ECHO consortium. And so that's a very exciting future direction. I was talking to Dr. Conway about this a little bit earlier, am interested in sort of risk stratification or subclassification in general. And so taking a different approach, instead of going deeper into molecular health or metabolites, but taking a step back and looking at what sort of routine EMR data are we collecting in the perinatal period that could inform subclassification, either in conjunction with metabolomics or separately. I've been doing some work in Dr. Jansen and Teresa Powell's lab this summer, looking at placental signaling, and so exploring how some of these associations result in protein expression using placental homogenate would be an interesting future direction, and allow me to sort of investigate new targets given what we see in blood in a different tissue. Lastly, I see the potential to explore subclassifications in pediatric type 2 diabetes. This is something I'm not overly familiar with, but I'm very interested in. And given that the in utero environment has such an important sort of potential programming effect on insulin resistance in childhood, really like to explore how maybe even differences in insulin physiology among kiddos with diabetes in youth are related to differences and complications in the future. And so although this talk was relatively short, I did want to just list a few different projects that are either ongoing or that have been completed, so that people get a sense of other things that I've done beyond the world of diabetes and pregnancy. And I've kind of grouped these on the right if they're more related to child health, and on the left if they're more related to women's health, and then kind of on the bottom if they tend to overlap. But with that, I want to thank you all for your time, and particularly Dr. Pern and Dr. DeBellia for just being fantastic mentors, and then also mentors as part of my T32, and Dr. Jansen for letting me work in his lab, and then also for putting up with my terrible scratchy voice. I really appreciate it, but I will take any questions, and thank you very much.
Thank you very much, Dr. Francis, for that very informative presentation. I don't know who has questions, but I will start off with one. No real right or wrong answer. A lot of your research focuses on categorizing people based on their metabolic phenotype, and the field of precision medicine is moving more and more towards individualized risk prediction and thinking kind of about things on a spectrum, especially if we think about developmental overnutrition, because it's unlikely that you have it or you don't, but it's really that there's varying degrees of it. So how do you see your work contributing to this paradigm?
Yeah, it's funny. I think it was Dr. Conway I was talking about a spectrum with earlier today. And I don't see my work contributing to personalized, individualized medicine. I see the work that I'm doing right now as sort of providing support for empirical data that we can sort of re-stratify how we identify risk in pregnancy. And so if you think of one end of the spectrum being using BMI, which is a pretty gross measure of metabolic health or even body composition, and the other being a very personalized individual approach. I'm falling somewhere in the middle where I think that we can do better than BMI greater than or equal to 30, but probably will lose quite a bit of efficiency if we take just a sole individualized approach.
Thank you. Are there other questions? I am armed with more just out of my own curiosity, but I think I should hold them. Well, Ellen, I am curious about the placenta. I have done some work prior to coming here to see you. And, one thing I have always thought about with the placenta is that you can't look at it until after pregnancy is over, and you can't know what's going on with the placenta really while the woman's pregnant. And so how do you view the placental piece in terms of identifying risk and what to do about it?
Yeah, I think for me, I'm going to not speak too much on this because I don't think I can do justice to the role of the placenta really in risk stratifying in pregnancy. But what I see it as an important piece of my work is validating the meaning of clusters. Because as I said, untargeted metabolomics, and I'm looking at all women in pregnancy. So your traditional approach would be to look at within a diseased group, are there sort of subclasses already there? And I'm saying, okay, well, I think right now we're missing some women who are at risk, who are being classified in that overt disease group. And to help, I think I'm using the word validate the meaning of these clusters. I'm using placenta data as a way to say, okay, we're not just seeing differences in circulating levels of amino acids, but we're also seeing differences in the phosphorylation of proteins in the placenta that are involved in amino acid transport. And maybe I will work with some very intelligent colleagues who then can take that information further and to identify maybe why that is, or when in pregnancy that might be most important. But I think that would probably be beyond the scope of, what I'm able to do.
Team science.
Yeah, absolutely.
Donna?
Yeah.
Hi, Donna.
Hello. I have several kind of questions, but they're somewhat different. One is more about your initial findings that you presented with the metabolomic clusters in children as they relate to what you call different forms of overnutrition. And your finding that were surprising to you, that there were more similarities than differences, and what that might suggest. So I think you said that that might suggest that there's more common
pathophysiological overlap. And I think that has implications for us as epidemiologists when we go to model this as an exposure.
That's what I wanted you to say.
Yeah, and we see this. So this really started to come up when I was at NIH looking at adipokines in relation to GDM, and then being asked to adjust for pre-pregnancy BMI. When what I think this metabolomics analysis has shown, at least in EPOC, is that adjusting for pre-pregnancy BMI when your exposure is GDM is adjusting for some of the exposure, because when you use BMI and GDM, yes or no, you're picking up on something that's very, very similar. Now, I tend to think of there being a lot of difference within those phenotypes, but I don't think BMI and glucose capture it. I think metabolomics in women is going to capture it, which is why I applied for the K, to use that data to say, no one else was surprised but me to find that there was a lot of similarity in obesity and GDM. But I'm sort of arguing that to pick up on the differences I'm interested in, I need different data.
And the other point that you made is very well taken. We need to educate the scientific community, the reviewers of various papers who continuously ask, why didn't you adjust for this or that?
Right.
That approach is simplistic and doesn't accomplish what they think you're asking for. The other question is totally different. And people could still go back to your presentation after I ask this question, but since this is a talk that is given in response to an application to a faculty position in a department of epidemiology, I wanted to ask you, what's your ideal workplace when you look at, the very nice next steps that you outlined? Where do you see yourself working to accomplish these next steps?
That's a great
question. You
question.
don't have to name names, you just can describe it.
No, I've thought a lot about the science that I want. Let me rephrase that. I thought a lot about the science that I'm being funded to accomplish currently, and a lot about the science that I'd want to do in the future that will build upon that, and what the environment looks like that will support that. And it, to me, is a strong funded perinatal interest, not just in OB, but in pediatrics and in epidemiology. And I was talking to Dr. Norris about this earlier, and it makes sense that this is a great scientific home for me because my science has grown so much because of the strength in reproductive science and obstetrics, the strength in pediatric health, and the strength in epidemiology. So I really see having innovators and the infrastructure and the funding to support science in those three interrelated areas as being a really important piece of where I go. Yeah.
Thank you.
Are there any other questions for Dr. Francis? Okay. If there are not, I am going to thank Dr. Francis for a wonderful presentation. Rebecca Conway says, great job.
Oh, thank you.
Then I will close this out and give folks an extra 10 minutes of their time back. And Ellen, I'll bring you that cup of hot tea.